Aloha, everybody. Welcome to our Slice of PI-CASC monthly seminar series, typically held on the first Tuesday of the month at noon here in Hawaii. I'm Brad Romine, the Deputy Director of the University Consortium of the Pacific Islands Climate Adaptation Science Center, or PI-CASC. I'm also a coastal scientist with the University of Hawaii Sea Grant College Program. We hope you've been able to attend some of the other wonderful presentations we've hosted this fall and winter. For anyone that's joining us for the first time, PI-CASC is a collaborative partnership between the U.S. Geological Survey and a university consortium comprised of the University of Hawaii at Manoa, University of Hawaii at Hilo, and the University of Guam, excuse me. This cooperative center is designed to support sustainability and climate adaptation in communities across the Pacific Islands. PI-CASC provides natural resource managers and cultural stewards in federal, state, and local arenas access to the best science available on climate change and other stressors impacting the region's natural and cultural resources. We emphasize cooperative efforts between researchers and managers aiming to create actionable science and accessible products to use on real problems. This Slice of PI-CASC seminar series provides a platform for sharing ongoing and state-of-the-art climate adaptation research and science to management applications in Hawaii, US affiliated Pacific Islands, and beyond. This presentation is being recorded and will be available on the PI-CASC website under news and events. We ask that you please keep your microphones on mute during the presentation. We will have time for questions following the presentation. Please feel free to leave questions in the chat along the way, or you're welcome to save them until the end of the presentation as well. At the conclusion of our seminar, we ask that you please complete a short survey to help us in planning future PI-CASC seminars. The survey will pop up after you log off of this Zoom meeting. It is my pleasure to introduce our speaker today, Dr. Casey Barton, who will be presenting Life on the Edge, Coastal Plant Resilience Under Sea Level Rise in Hawaii. Dr. Barton grew up in Colorado, has an undergrad biology degree from Stanford University, did post-baccalaureate research as an intern at Wesleyan University on plant drought tolerance. She earned a PhD in ecology and evolutionary biology at Colorado University Boulder, investigating plant herbivory defenses. She also did a NERC fellowship for postdoctoral research at Royal Holloway, University of London, including field research in Finland. Casey joined faculty at UH Manoa in 2010, was part of the botany department until 2020, when that department merged into the School of Life Sciences. Since joining UH Manoa, her research has focused on two main questions. Are island plants fundamentally different from continental plants? And how do plant function and stress tolerance shift as plants grow up? In her lab, she's examined many different stressors, including drought, salinity, fire, competition, and herbivory. She enjoys exploring Hawaii's beautiful natural areas with her family, including her 14-year-old son and 10-year-old daughter. Casey, I'll pass it over to you. The Zoom link is yours. Mahalo. Thank you very much for joining us today.
Great. Thank you, Brad, for the introduction and the invitation. I'm very happy to be here. I'm going to share my screen. All right, thanks everybody for showing up today to hear about our work in coastal dune plants. I'm really excited to share this work today. I actually got involved in coastal plant ecology just by accident because of the interests of some fantastic students working in my lab. And it was because of their cool insights and discoveries that coastal plant ecology is now one of our main research foci. So I'm really happy to get to share some of their exciting work with you today. And it's led to the project that I'm really going to focus on today about salinity tolerance, which we're doing in collaboration with people at National Tropical Botanical Garden, Shauna Walsh and Dustin Wolkes, and also Maui Nui Botanical Garden, Tamara Sherrill. So thank you so much. These are, oops, sorry. These are the systems that we're working in, the coastal dune, also called coastal strand, and the plants that live in these places. So these are really beautiful habitats, and the plants that grow in these habitats are fascinating. And they're also super important. So they're ecological and ecosystem engineers, that they provide a lot of stabilization of these coastal dunes. They create structure, they buffer inland habitats from storm events and big waves. They're also important for biodiversity, not only because of the plant species that grow in these areas, but also because they provide critical habitat to associated species such as nesting seabirds and endemic insects like the Hylaeus bees. They're also culturally important. We use these habitats for our own emotional and social well-being. We recreate here, we practice cultural traditions. And so the persistence and stability of these ecosystems is really fundamental for the islands. And this is important because these are really narrow and small ecosystems. They're really just a slender corridor along the coast, and yet they play these really important roles. So we first got involved in coastal plants because of the interest of an undergraduate student, Kelsey Coleman, about a decade ago. And Kelsey was interested in the endemic parasitic dodder. And in particular, she did this beautiful anatomical work characterizing, as you can see in this image here, the hostorial connections between the dodder and the host plant, the native dune plants that it parasitizes. You can just see the dodder growing down here in the base of this photo. So in collecting the specimens for her anatomical work, Kelsey and I visited sites across several different islands where they had previously been described from the herbarium records at Bishop Museum. So we went back to these sites, we collected specimens, and then she came back and did all this great anatomical work. And in doing this anatomical work, she actually discovered that the non-native congener, Cuscuto campestris, is also established in some of these coastal dune sites, which was a surprise to us because usually we see the non-native in inland sites where it parasitizes non-native host plants. So it was surprising to find that the non-native had actually moved into these dunes where it was parasitizing native dune species, like what the native dodder does. And it would be really easy to miss that this non-native is established in coastal dunes because the trait that differentiates the native and non-native are these really tiny scales at the base of the stamens. So it's not something you can easily differentiate just based on morphology. So on the one hand, it was really disappointing to find this non-native species in the beach, but it also, I suppose, wasn't that surprising, given that non-native plants are a known major threat to native plants across all habitats in the islands, and particularly in lowland habitats like these coastal dunes. So what you can see from this map here is that a lot of the lowlands and around the coast are this dark blue, which are the non-native dominated habitats, and that usually you have to go up to higher elevations to find mixed or native dominated plant communities. And although this is a general pattern, and I think well characterized by this map here, I want to highlight that the coastal dunes that we study probably actually aren't even visible on these maps because they're so narrow. And in fact, if you go to many of these coastal dune sites across the islands, there's still a high density and diversity of native plants. So this seems like a bit of an outlier or an exception. These are lowland habitats still largely dominated, at least in many places, by native species. So this got us wondering what was going on in these systems that allowed for the native diversity to persist, given all of the disturbance and introduction of non-native plants that we suspect is probably happening. So in general, the dynamic between invasive and native plants has a lot to do with the functional overlap between native and non-native species. So the characteristics of the invasive species, but also the characteristics of the native species and native habitat where those invasive plants are trying to colonize. And this functional overlap has been by Darwin characterized as a conundrum because on the one hand, we may predict that there's a lot of functional overlap because in order for those invasive plants to become established, they must be pre-adapted already to those conditions to get through any kind of environmental filters that would otherwise exclude them. So we predict them to be similar to the native plants. But on the other hand, we also predict them to be different, because if they're too similar to the native plants, then there could be biotic resistance or competitive exclusion that would prevent them from becoming established. So here's the conundrum. Are the invasive plants similar or not similar to the native plants where they're establishing? And of course, there's a huge disclaimer here that there are some really problematic invasive plants in coastal dune ecosystems, such as kiawe and ironwood. And I appreciate this photo down here that shows kiawe with a toilet, because that's basically how we think of it. So in thinking about the functional overlap between native and invasive plants, we characterized functional traits associated with growth strategies, potential competitive interactions, such as photosynthetic traits, leaf morphology, leaf chemistry, of 26 native and 27 invasive plant species and 2 sites on Oahu. And this was a project led by a graduate student in my lab at the time, Kari Bogner, who got interested in coastal dune restoration because of an internship she was doing at Bellows Beach. And she and Patty Polly and Nick Nguyen and I collected these data. And then postdoc Andrea Westerband and another graduate student, Tiffany Lum, came on later to help us analyze and interpret these data. So I don't want to go into a lot of the details of each of these different figures, but what I'm showing you are the pair comparisons for the exotic or the invasive species and the native species across all of those traits that I had listed on the previous slide. And the main conclusion is that there were no trait differences. So functionally, the invasive plants establishing in these coastal dune habitats are really similar, indistinguishable functionally from the native plants. And this was pretty interesting and different from what happens in a lot of other sites around the islands. So on the one hand, this could mean that there could be a lot of competition occurring between the native and invasive plants, and potentially the native plants are maintaining their dominance due to their competitive abilities. Or on the other hand, it probably also highlights the importance of the environmental context and how the environmental conditions of these coastal dunes are really specific and actually filter out most species that are not pre-adapted, so that the ones that successfully establish look functionally very similar to the native plants already occupying these sites. And I think there's probably a lot to that, because if you think about these coastal dune habitats, they're super stressful. This is not an easy plant, not an easy place for a plant to grow. There's a lot of solar radiation, which leads to light and heat stress. They tend to be very water limited because of not very much precipitation into these habitats, although they get a lot of water exposure from seawater that has high salinity. They tend to be very windy. There's a lot of physical disturbance because of sand abrasion, trampling, waves, and storms. And all of this means that there's probably strong environmental filters, allowing for only a very narrow type of functional strategy to be successful in these particular conditions. Now, because these are historically stressful habitats, we do expect that the native plants and now these invasive plants have adaptations that allow them to cope with these stressors. The problem, and the reason that we're researching this now, is because human activities are totally exacerbating all of these stressors. Climate change and our increased densities and activities in these coastal dune habitats are making these environments much more stressful than they have been historically. And we don't really understand how this alteration to the conditions is going to affect the persistence of these coastal dune plants, and potentially whether it could tip the balance in favor of the invasive species, allowing for them to increase their densities and potentially displace the native plants that we still see common in these areas. So this is what we've been currently pursuing, specifically how does climate change influence the functional strategies and performance of these coastal dune plants. And to understand this, I think we just need to spend a moment looking at the ecohydrology of these systems and remember that coastal dune communities occur above the high tide zone. So these are freshwater systems. The plants have roots that go down and access this fresh groundwater, and they depend on that fresh water for all of their function and survival and growth. They are, of course, because of their proximity to the ocean, regularly experience salt stress, and this can occur both above ground because of regular salt spray, because of the winds coming off of the water, and also from large storm events. So there's a regular kind of exposure of salt ions above ground. And of course, there can also be exposure below ground due to saltwater intrusion. And this is becoming particularly problematic under sea level rise. As the ocean levels rise, and that's going to replace the fresh groundwater that these coastal dune plants depend on. And then exacerbating all of this are changes in the precipitation regimes. So under historical conditions, although there was regular deposition of salt ions from salt spray or big storm events, the rainfall would dissipate the accumulation of those salt ions from the surface sand, allowing for them to be reduced and the plants to then have access to fresh groundwater for their function and growth. But climate change is not only increasing the deposition of salt into these ecosystems, but it's also reducing the amount of rainfall. And that means that the salt ions are accumulating far beyond their historical levels. And so this is our concern in what we're investigating is how do these new novel salinity regimes in these coastal dune habitats affect these plants? And there's been a lot of work about salinity stress in general, and we know that it can affect plant function both directly and indirectly. So some of the direct effects are things like salinity can cause protein denaturation, chemical imbalances by displacing ion transporters. It can cause cell death because of cell membrane destabilization. But perhaps most importantly is that it can cause osmotic stress. That's kind of an indirect effect. And basically what happens is the salt ions accumulate in the soil that reduces the soil water potential, which makes it harder for the plants to access water, and they experience osmotic stress similar to what would happen under a drought or water limitation. So these plants become really water limited under high salinity. So to investigate whether plants can tolerate salinity, we can define tolerance in general, stress tolerance in general, is the maintenance of fitness under a stress. In our case, that's salinity. And we mean fitness in the biological sense. So we would quantify that by measuring survival, growth, reproduction, and germination of plants exposed to stress or high salinity compared to plants growing under control or low salinity conditions. So that's our question. Can these coastal dune plants tolerate salinity? On the one hand, knowing that these are freshwater systems, we would predict that they probably cannot tolerate the enhanced or elevated salinity that's being caused by climate change, even though they are likely able to tolerate low levels or low durations of salinity exposure from salt spray. And our second prediction, I don't think we have any particular reason to expect that invasive species are more salt tolerant than the native plants, but we certainly want to know the answer to that. So our second prediction is just that there's no difference, and it's something that we can address. All right, so the first examination into this was led by a graduate student in my lab, Tiffany Lum, who designed some really elegant experiments to investigate salinity tolerance of two common and widespread coastal dune plants. And she tested their salinity tolerance across their whole life cycle. So across all of their ontogeny, including seeds, seedlings, juvenile plants, and flowering adult plants. And then for more mechanistic insights into how the plants can tolerate salinity or not, she collected functional trait data throughout all of these stages, including photosynthetic traits, morphological traits, and some anatomical traits. And just to sort of share some of the highlights of her work, she found that salinity caused a lot of stress, particularly in those early life history stages. So she saw that salinity reduced and also delayed germination. Plants that were exposed to a pulse of salinity at the seedling stage not only experienced reduced growth during the seedling stage, but those effects actually persisted all the way through their life cycle into the adult stage, leading to delayed and reduced flowering. In contrast, for plants that were exposed to a pulse of salinity at the juvenile or adult stage, she observed very little effects on their growth or survival. So there seems to be this increase in salinity tolerance as plants grow up. And because of all the great functional trait analysis that she did, she found that there were two traits that emerged to be really important indicators of the mechanisms of salinity tolerance, and those are chlorophyll content and water use efficiency. So the older stages that had higher water use efficiency and greater leaf chlorophyll content were also the ones that were able to grow the best under salinity as evidence of their salinity tolerance. And I think the water use efficiency really highlights the importance of the osmotic stress and the water stress that plants experience when they're exposed to high salinity, such that those plants that could photosynthesize the most while maintaining the most water balance or minimizing water loss are the ones that were able to perform the best under salinity stress. So this is a really important insight because often in the field, we observe mature plants and we focus our investigations on the adult plants that we can easily see and measure. And those plants turn out to be pretty salt tolerant. But it's easy to miss these little guys, the small ones, the seeds and the seedlings. And that's unfortunate because population persistence depends on seed germination and seedling establishment. And without successful seedling recruitment, eventually, when those mature plants die, there will be no young plants to replace them, and you can end up getting a really abrupt and dramatic crash in a population. And so we really do need to pay attention to these early life history stages that seem to be the most sensitive to this factor of climate change and whose function is absolutely essential for population stability. So moving on to our current work, we wanted to know then, are these observations of low salinity tolerance at the seed and seedling stage generalizable, or were they perhaps really specific to those two species that Tiffany Lum studied? So our current work is really expanding this now to include as many species as we can test, focusing in now just on those seed and seedling stages. And this is the current project, and this is the one that's funded by Sea Grant and PI-CASC, and involves this wonderful collaboration with folks at Maui Nui Botanical Garden and National Tropical Botanical Garden. And it's been a whole lot of fun. And we've had lots of wonderful students and postdocs and volunteers help out with this. And it's really great honor for me to be able to present our work today. All right, so to investigate seedling salinity tolerance, again, we're using an experimental approach in the greenhouse. And here's the basic experimental design. We start with seeds, which are collected from the field by Maui Nui Botanical Gardens. We germinate those seeds, we grow them up. We're really interested in this seedling stage, but as a basis of comparison and to see if species generally increase in salinity tolerance as they grow up, we are also testing the juvenile stage, which is down here. So as plants reach those stages, they experience then a 3-week seawater treatment period where Anna McCormick, who's really running these experiments, waters them every other day with seawater. And at that point, then they stop getting treated with seawater and they undergo a 3-week recovery period where she waters them with fresh water again to allow them to recover from that stress. And then we harvest the plants so we can measure their total biomass and growth. And although we would prefer to do this with actual seawater as we've done in the past, because of the pandemic, we weren't able to access our sources of seawater. So we've used instant ocean or synthetic seawater for these experiments. For the seed salinity tolerance experiments, those are also done using an experimental approach by our partners at National Tropical Botanical Garden. And they've been able to use actual seawater. So you can see Shauna Walsh here collecting seawater in the field. They then expose the seeds to the actual seawater, monitor germination. And then once there has been no more germination under the salinity treatments, they also rinse those seeds to look at recovery. And that allows us to assess whether the salinity actually kills the seeds or whether those seeds can persist through the seawater treatment and germinate after they are exposed to fresh water. And our goal here, like I said before, is really to try to identify general patterns. So we've been including as many species really as we can get access to and for which we have time. And this includes a lot of common and widespread species because they're really the foundation of these coastal dune communities. But we have also included a couple of endangered species and of course, some of the key players of the invasive species that we know to be problematic in these dune habitats. All right, so here are the germination results. So I realize this is a lot of panels and very small text. On the bottom axis, the x-axis, you can see the different treatments of salinity. So 100% seawater is about 35 parts per thousand. And then we have diluted treatments that were also used at 10 and 20 parts per thousand. On the y-axis is percent germination. And within each of these panels are the results for an individual species. The light gray bars indicate the percent germination under, for zero, the control conditions or these different salinity treatments. And then the filled or black part of the bars indicate the recovery germination following that freshwater rinse. And we don't need to go over each of these panels. The take-home messages here are, first of all, it's really variable among species, which is interesting because all these species co-occur in these coastal dune communities. And yet they're not responding to salinity in any kind of uniform way. That's interesting. Second of all, we see that for all species, there's very low or no germination under the full seawater treatment, indicating low salinity tolerance and a definite sensitivity to salinity and seawater for germination of these species. Some of them could recover quite well when exposed to freshwater and achieve high germination, and some of them could not. So it appears that the seawater actually killed those seeds and they weren't able to recover after exposed to freshwater. Some species had pretty high tolerance and high germination under low salinity or the diluted seawater treatments. So there's a lot of variability here, but in general, we see sensitivity to salinity across all species, confirming that Tiffany's results are generalizable, at least across the species we've been testing. For the seedling and juvenile results, I'm going to show you all of those results in this kind of format. So I've calculated effect sizes for each species, which is a relative response of the salinity treated plants to the control plants. And that allows us to assess these patterns across all the species at one time, even though they differ in their overall size. For all cases, if there is a difference of 0, that indicates that the plants have similar performance, either survival or growth under salinity or control conditions. That's indicative of full tolerance, right? No difference in growth in salinity compared to control. If a plant is sensitive to salinity, we expect there's a negative number. That's a reduction in growth or survival in salinity compared to control. And that would be indicative of a glycophytics strategy. And on the other hand, for some species, they may have a positive effect size, which would indicate actually enhanced survival or growth under salinity compared to control conditions. And that would be indicative of a halophytic strategy or a really salt-loving species. But as we go from high to low on the y-axis, that goes from high tolerance to lower tolerance, a greater suppression of performance in salinity compared to control. Oops, and there we go. That's our first result, seedling survival. And what you can see up here at the top is quite a few species that had no difference, meaning 100% survival in salinity and control conditions. That indicates that they're at least able to survive through the salinity treatments. And then we also have some species where 100% of the individuals in salinity died compared to controls, no mortality in control. So those are very salt sensitive species where they cannot even get through that 3-week salinity treatment. And then some species here in the middle range showing intermediate salinity. The yellow dots are the invasive species and the blue dots are the native species. And you can see that there are a couple invasive species with high tolerance and one of the invasive species with low tolerance. So again, there's definitely variation across species ranging from complete survival under salinity to complete mortality. This is the seedling stage. Here's the survival data for the juvenile stage. Again, in fact, we have more species now where all of the individuals survived that salinity treatment, indicating high tolerance, at least with the metric of survival. And a few species, again, that had 100% mortality at the juvenile stage that absolutely cannot handle these salinity treatments at the juvenile stage. And a few species intermediate here. So again, species variation. Here are the results now for growth at the seedling stage. So these are the plants that were exposed to that salinity period at the seedling stage, and the ones that survived so that we could measure their growth at the end of that recovery period. And what you can see is that across all of these species, they're all negative, indicating less growth for plants under salinity than control conditions. So none of them fully tolerated the salinity treatment in terms of growth, even though they had that 3-week period of fresh water to catch up again to the control plants. None of them were able to achieve that. The most salt tolerant, the one with the least suppression of growth, is Aveo. Here's Kiave, the most salt tolerant among the invasive species, and all the way down here showing about a 90% reduction in growth under salinity compared to control is our least tolerant species at the seedling stage. But look at this spread. There's a ton of species variation, again highlighting how these coastal dune communities consist of a lot of functional diversity. They're certainly not all adapted to the salinity stress. They have adapted to different degrees. Here's the juvenile growth. This looks pretty different, and that's because we actually can see this 0 threshold, the ones that would indicate full tolerance. And we have 2 species, a Kuli Kuli and a Veo Veo, that actually have higher growth under the salinity treatment than control at the juvenile stage. So that suggests that those 2 species are achieving a halophytic strategy at the juvenile stage, such that they actually grow better when they're exposed to salinity than when they're exposed to control conditions. So that's pretty interesting. And neither one of them was halophytic at the seedling stage. So this again highlights how ontogeny is really important, and what may be sensitive at the seedling stage can turn out to be much more tolerant at the juvenile stage, just a couple weeks later in their development. And then all the other species have suppressed growth under salinity compared to control, again identifying them as having low salinity tolerance. Here's Kiave, again, the most salt tolerant among the invasive. And interestingly enough, ironwood was the most susceptible to salinity at the juvenile stage, with the greatest reduction in growth under salinity compared to control conditions. If we synthesize across all of these species now, we can see some of the general patterns. And I'm showing you that here, which I've analyzed using meta-analysis. Here's the 0% change. And there's a significant difference across all of these species, such that the juvenile stage is on average more tolerant in terms of biomass than the seedlings. So the juveniles on average experienced about a 30% reduction in growth, while the seedlings experienced about a 65% reduction in growth. Again, confirming Tiffany Lum's observation that those early stages are more vulnerable to salinity tolerance, and that for many species, they achieve greater salinity tolerance as they grow up. Another interesting and pretty exciting result is that across our species pool, the native species are on average more salt tolerant than the invasive species. So in both cases, we're seeing a decrease in growth under salinity, but an even greater suppression of growth for the invasive species compared to the natives on average. But of course, we've only included a few invasive species, although they are some really important ones. And so we might want to expand our species pool to confirm this result. All right, there's a lot of species variation, and that's interesting, but it also makes us wonder what's going on and what's driving this species variation. So I just want to share one functional trait response with you that we think is really important, and that is stomatoconductance. So in case you haven't been reviewing botany lately, stomata are the small pores on the leaf surface, and they regulate gas exchange in and out of the leaf. Of course, the gases that are going out of the leaf are water vapor. So this regulates transpiration, or the evaporation of water out of leaves. It also regulates diffusion of CO2 into the leaf, which is important for providing a CO2 supply for photosynthesis. Plants can regulate the aperture of stomata, so they can close the stomata under stress to reduce the loss of water, primarily through transpiration. But the cost of reducing gas exchange to maintain water is that it also constrains the amount of CO2 that's going into the leaf for photosynthesis. And that's how water stress can then lead to reduced growth. It's because of this constraint of CO2 uptake in order to maintain water inside the leaves. So we can measure the rate of gas exchange in and out of stomata using a parameter. And that's the result that I want to share with you. So here's for the seedlings and this for the juveniles. And we measured stomatoconductance right at the end of the salinity period treatment on the last day of that 3-week salinity treatment. And then we also measured it again at the time of harvest after they had had 3 weeks of fresh water to recover. And as expected, at the end of salinity, all of the species show a reduction in stomatoconductance at the seedling stage. This again confirms that osmotic stress is an important consequence of salinity. So they're water stress that close the stomata, and that leads to a detectable decrease in stomatoconductance in salinity conditions. The interesting thing is to look at how these species respond during that 3-week recovery period. So importantly, some of these species that had even a great reduction in stomatoconductance respond really well. And they increase the stomatoconductance again during that 3-week recovery period, and even in some cases achieve greater stomatoconductance than the control plants. On the other hand, some of these species, you see it's a flat line, and that indicates that that decrease in stomatoconductance that occurred at the end of salinity is persisting even through that 3-week recovery period where they're getting fresh water every single day. And it's this persistent stress, this lack of stomatoconductance, that is likely leading to that reduced growth that we can detect in the biomass data. Same story in the juveniles, right? So a suppression of stomatoconductance at the end of salinity for all species except for one, Milo, which you can see is actually right about at 0, indicating that they're not doing any stomatal regulation under salinity, suggesting that it's very tolerant of water limitation. And then the same sort of pattern where some of these species recover really well and achieve high stomatoconductance during that recovery period, and some species that just can't recover and they have low stomatoconductance even after 3 weeks of fresh water. So if we want to see whether this kind of dynamic of the recovery of stomatoconductance can predict the biomass results, we can do a regression. And so what I'm showing you here is how that effect size of the percent reduction in stomatoconductance at the recovery period predicts the biomass response of those species. So each of these dots is a species, and I'm showing you for the juvenile plant stage in the yellow, and the blue line is the seedling, and they actually have the exact same slope. In both stages, those species that achieved a higher stomatoconductance, so a less decrease in stomatoconductance during the recovery period, are also the ones that had the least reduction in biomass. So there's a positive relationship between the recovery of stomatoconductance and the salinity tolerance detected as biomass. Again, underscoring the importance of osmotic stress underpinning the salinity stress that occurs during sea level rise or other kinds of salinity treatments. And that was detectable at both stages. All right, so these experiments that I've just described actually are simulating this kind of chronic, persistent exposure to high salinity over like a 3-week period. And we were also wondering that under sea level rise, there's going to be more big storm events, more coastal flooding events like this. And so we wanted to try to simulate that as well using an experimental approach. And the reason that this is important and actually different from the kinds of treatments that we've previously been doing is that inundation is not just exposing those plants to enhanced salinity, but it's also creating an anoxic below ground environment, right? So it's causing oxygen deprivation to the roots, and that can cause root dieback, and that exacerbates this osmotic stress that the salinity causes. So we did these inundation experiments to try to simulate that sort of coastal flooding event that's happening increasingly due to climate change. And I say we, but I really mean these fantastic undergraduates in my lab. So this here is Mai Lei Wang. And for her honors thesis, she's pursuing these kinds of inundation experiments. And she's doing it both in the greenhouse, but also in the field. So here she is on Kauai at National Tropical Botanical Garden, where last summer she set up these inundation experiments in the field. And then she's also doing them right now in the greenhouse. And then there's another student, Devin Montesinos, who for her GES thesis is doing another different set of inundation experiments in the greenhouse. And both of them are really working hard and have tons of data, but haven't yet analyzed that. So those are still pending. And then finally, we've actually used this same kind of inundation experimental approach for an outreach program that Anna McCormick and I are currently running at Kalani High School with 3 different marine science classes. And we've helped the students set up these inundation experiments right directly in their classroom. And the students are running the experiment themselves. Anna and I go back every couple of weeks and we help them do things like measure traits and height and think about how to analyze the data. And this has been really fun and is a great opportunity for these students to practice the scientific method, learn about native plants and climate change. And it's been a really fun experience so far. Also in progress, so we don't have those results yet either. So clearly, what we've been finding is low salinity tolerance across these coastal dune plants, particularly at the seed and seedling stage. And this has got us thinking, is this something specific to our plants here? Maybe these are island plants and they're particularly vulnerable to climate change for some reason. And so we wanted to see how they fit in with coastal plant responses to salinity in a global context. So right now, Anna McCormick and I are pursuing a global meta-analysis, and we're being very inclusive to include all supertidal coastal ecosystems. These are all the ecosystems above the high tide zone, so not salt marshes or mangroves, but systems like dunes, forests, plains, and cliffs. We're including tolerance responses such as the ones that we've been measuring in our experiments, like biomass, survival, reproduction, and germination. We're also really interested in the mechanistic responses, the ecophysiological responses of plants to enhance salinity. So we're also collecting data on trait responses involved in photosynthesis, hydraulic function, leaf morphology, leaf and stem anatomy, and leaf chemistry. And like I said, Anna and I have been working on this, and in the early days of it, we got some great contributions from Taylor Young, who was a SURF intern back in 2020. So this is a really big project. We're still doing it, but so far we have data extracted for 136 plant species, collecting data from 87 publications published between 1988 and 2020. And our data set so far includes 3,490 data points of salinity tolerance. And that includes trait and tolerance responses to experimental salinity, like what we're doing, but also natural salinity variation in field conditions. So I pulled out the data points so far from the global meta-analysis that just include total biomass, because that's similar to the results I've just been showing you. And what you can see here, that's 156 data points for the meta-analysis. And we see the same kind of pattern that we've been seeing in our experiments and that Tiffany Lum first observed. And that is that at the adult stage, there's no detectable effect of salinity on growth measured as total biomass, right? So this error bar here, this confidence interval overlaps with 0, meaning no change or full tolerance to salinity. But just as we've observed, early ontogenetic stages have reduced salinity tolerance. Juveniles experience globally about a 40% reduction in biomass, and seedlings are even less tolerant, about a 55% reduction in growth. Here's the results I just showed you from our current experiments for a basis of reference. And you can see they're similar. There's actually a greater magnitude of difference in our species between the juvenile and seedling, but the same general pattern. And this is really interesting. And it certainly confirms this observation that salinity tolerance increases as plants grow up, which is great news for the persistence of adult plants under sea level rise. But it's certainly not good news for seedling recruitment. And for population persistence, we're going to have to be really careful to monitor and pay attention to those seedling dynamics because they're much more sensitive to enhanced salinity. All right. One of our goals is to make this data and these results accessible and available to practitioners who are working in the conservation of these systems and restoring degraded dunes and may be able to use these estimates of salinity tolerance. So what we have in development right now is this interactive web tool where we can present our salinity tolerance indices, those effect sizes that I've just shared with you. And these can then be retrieved by anybody who wants to get them from our website. And I'm showing you this. The metric here goes from 0 to 100, and I've been showing it to you as 0 being high down to -100, but it's conceptually the same thing, just on a slightly different axis, and it's measured the same way as that log response ratio effect size. So I want to just show you a couple of key features of what this tool is going to include. And I'm going to do that by taking advantage of another tool that I have on drought tolerance index. So it's going to be conceptually the same, only of course, right now, what I'm showing you is the features of the drought tolerance index, but it'll be the same thing where you can select a species of interest from a pull down menu. And after you've selected the species that you want to have these data for, then it shows up here with the effect sizes reported here on this measuring stick. And if we have data from multiple populations, as is shown here for acacia koa and drought tolerance, both populations are going to be displayed on the tool. And if you hover over one of these, it would then display the source population that those seeds came from. So you could actually relate it directly to a field site. And you could also include all native species, for example, and display them all simultaneously. And this allows you to see the spread of drought tolerance that we've been observing in our experiments. You could then identify what's the most drought tolerant species, what's the least drought tolerant, or in our case, salinity tolerant species. And that could help with conservation by knowing which species are the most sensitive to the stress and which ones are perhaps the most resilient that we could prioritize for initial outplantings. And then we don't have it shown here, but ultimately this is going to have a map so that you could, again, go to the source population, find out the site where those seeds came from, and that would give you the information about where the data relate to. Okay, so the final take-home messages, I think, are that what we've been seeing is that most coastal dune plants are not salt tolerant. So despite their proximity to the ocean and their likely regular exposure to salinity, they haven't evolved salinity tolerance throughout all of their ontogeny. And they're particularly vulnerable during those early stages of seeds and seedlings. And this is really important because that could create a bottleneck for population persistence. We don't observe that invasive plants are more salt tolerant, perhaps even less salt tolerant than the native plants, suggesting that perhaps the dynamic between those native and invasive species is unlikely to change under increased salinity from sea level rise. But I'm hesitant to draw too strong a conclusion about that, given that we only included a handful of invasive species so far in these tests. So I think careful monitoring and careful treatment of these early life history stages during restoration is going to be critical, because obviously when restoring or conserving natural communities, natural seedling regeneration is one of those end goals. And that's going to be really constrained by increases in salinity, which would be easy to miss if we're not really paying attention. And again, just to emphasize how tiny these plants are, and it's really easy to miss them if you're not looking carefully when you're out there on the beach. And then future direction. So this work we've been doing for several years now, but I really feel like we're just getting started. We have some exciting results that have just led to more and more questions. So some of the directions that we're interested to take this include the incorporation of more population sampling within species. So far, our strategy has been to include as many species as we can, because we want to try to characterize these general patterns. But of course, for most plant traits and functions, there's considerable within species variation. And that could be really important for informing conservation and restoration that want to know about particular populations. So I think increasing the sampling of populations within species, especially widespread species, is going to be really useful. I also think that it's important to do more field experiments. We're ready now to really transfer this into the field context. There's obviously a lot more complexity and a lot of other stressors that are occurring simultaneously in the field. And because of potential interactions and non-additivities among multiple stressors, including abiotic, but also biotic stressors, means that potential tolerance, which is what we can quantify under these controlled conditions, may not necessarily inform us about realized tolerance that occurs under all of these other complexities. So that requires some field experimentation, and we're really excited to do more of that. And I think that's a main future direction. And then finally, of course, we want this information to be useful to people who are working on these beaches. And so if there are ways to increase communication, facilitate use of these data, add new species or populations of interest, we are very happy to have those conversations and adjust our approach to make it as useful as possible. So with that, mahalo, I appreciate the invitation and your attention, and I hope I have some time for questions.
Yes, we do. Thank you very much for that really interesting presentation, Casey. We do have about 10 minutes for questions if you can stick around that long. So
great.
Yeah, excellent. I'm not seeing any questions in the chat at the moment, so I just welcome folks to go ahead and raise their hand or chime in if I should miss your hand. Or feel free to enter in the chat if you don't want to go ahead and speak up. I do see, okay, see one coming through in the chat now. I'll go ahead and read. I think you can see it too,
okay.
Casey. But, yeah, what a great talk. The comment, what are the relative abundances of native versus exotic plant at your dune sites? Do you think these exotic species are driving their invasions onto these dune sites or taking advantage of native species decline?
Yeah, so that's a great question, totally relevant. We haven't been doing any community assembly work. That's not the kind of work that I do, but I think other folks have done some really nice estimates of densities, and it's highly variable, of course, across different coasts and different sites. Where we've been working, there's a really high abundance and density of native plants. And so in our particular sites, I don't think it's that there is a decline first of native plants and then the invasives move in, but we've also been targeting sites with lots of native plants to sample. So I think there's definitely interplay there. The functional trait analysis indicated that the plants that are establishing in the dune communities that we've been sampling are functioning really similarly to the native plants. So I think there's this pretty strong environmental filter that allows invasive plants that are pre-adapted to these stressful conditions to establish. And so that's a different kind of dynamic from a really different or unique invasive plant that's taking advantage of an open niche. That's not what we're observing in the species we've been focusing on.
Great, Casey, thank you. I see a comment to that question from Rebecca Ostertag in there.
Yeah. Yeah, Becky, I totally agree. It'd be great to take this functional trait approach and combine it with community assembly and go from there. We haven't done that because I'm not a community ecologist, but I appreciate that there are other people doing that well, and it'd be really nice to collaborate with them. Like you.
I see a hand up from Jim Patemra, and then a question from Rachel in the chat. Go ahead,
Yeah,
Jim.
great. Thanks, Casey. I recognize your background there.
Oh yeah.
It's kind of off, maybe off your central focus here, but in some of the places in the outer Pacific, like in the Marshall Islands, they're looking at using some of these coastal plants to actually keep the shoreline in place,
Mm-hmm.
like the Naupaka. I'm wondering if that's if you guys have thought about this balance between what might be a native species and what might be something that actually helps preserve the shoreline and the plants there. Thanks.
Yeah. Well, Naupaka's native here, but not there is what you mean. Yeah. Well, so I mean, my position is that we should prioritize native plants whenever we can. And I'm pretty hesitant with the idea of introducing new species for ecosystem function. I do like the hybrid approach where people have taken advantage of, particularly in our case here, the Polynesian introduced plant species. The ones that aren't invasive make great components of restoration efforts. And so I don't know. I mean, I think maybe some people have told me that Milo, it's not clear, maybe a Polynesian introduced. So that would be an example of that, certainly being used in restoration. But then there's the other sort of end of the spectrum, like How, which is pretty invasive, and I think is probably not a great species to be outplanting in restoration, even though it's a canoe plant. So I think we have to tread kind of carefully in that regard. And if there are sort of culturally important, non-invasive, but non-native species, then yeah, I think we're to the point where we can take advantage of those. But I'm not in favor of introducing new species that we know nothing about for that.
Right, great. Thank you.
Yeah, thank you.
Casey, I see a question in the chat from Rachel Lentz at PI-CASC. And it's something I was thinking about too, you know, from those of us that are involved in dune restoration projects, revegetation. Coming out of your work, do you see any practical way to assist seedling survival in dune restoration in dune settings?
I mean, the obvious answer, which of course everybody already knows, is to water them, right? I mean, the stress is the salt ions accumulating in the soil and the lack of rainfall to dissipate those salt ions. So when we water the plants in, it's not just because that reduces sort of drought stress, but it's also because it reduces the salinity stress. People know this. They often water plants in when they're out planting. The problem is, if we want a more hands-off approach, then that's not something that we can use in the long term. And if we're talking about natural regeneration, then we're not watering. So this is really tricky. And I think it's hard to consider sort of, I mean, admitting that you're basically going to be creating a system where you're watering all the time to enhance seedling recruitment that you don't know if it's happening or not, but you hope so. So you water during the time when seed production is happening. I don't think folks want to be doing that. In the long term, my guess is that we're going to have to do some assisted migration inland, because with climate change and sea level rise, we're going to be losing these coastal dunes over time. There's going to be too much seawater intrusion below ground. So I think it's going to be requiring a lot of management, is my guess. And I think it's important now to recognize this and start doing things like seed banking common species, so we can protect the biodiversity, that we can sort of anticipate that assisted migration is going to happen down the road. And that was one of our goals, actually. And the partners at Maui Nui Botanical Garden were able to bank a bunch of common species through this work. So I think that's really exciting and, you know, a way to sort of be planning ahead.
Thank you. We have time for one more question. I see one from Dr. Yunfin saying, when comparing the seawater period and recovery period, the responses of seedling versus juvenile of the same species seem to be inconsistent. And not all juvenile are consistently recovering better than seedling. What would be a good interpretation of that, to that, or any implications? Pardon me. Hopefully you can see that in the chat too.
Yeah. Thanks, Yin-Fan. That's a great question. And I sort of glossed over all that because I was highlighting the most tolerant or the least tolerant. But actually, there's quite a lot of agreement between the stages. So even though it wasn't necessarily the same species that sort of turned out to be the most tolerant, they were usually the sort of top three and bottom three kind of thing. So the absolute ranking wasn't consistent, but the general area is. And this is data that we're just starting to analyze. So of course, moving forward, we're going to be describing these as trajectories within species, so we can properly assess that. I think there's a strong consistency across stages, but it isn't perfect. And that's actually pretty interesting and greater complexity that we have to be thinking about some species more at the seedling stage and some species more at the juvenile stage. Yeah, good insight. You caught that.
Well, we're at the hour. I think we'll have to stop there. But please join me again in a round of applause for Dr. Barton. Thank you, Casey, very much.
Thanks, everybody.
Really appreciate it. So please join us at our next Slice of PI-CASC seminar. Please continue to come. The next one will be Tuesday, April the 5th. These are monthly on the first Tuesday. Information on that seminar and sign up information for that event will be coming soon. Or you're welcome to visit the PI-CASC website under events for updates on the seminar series. We also, again, please ask you to take a minute or two to complete a survey after you log off of Zoom. And thank you again for joining us. And we look forward to seeing you again in April. Mahalo.